So today we'll talk about bacteria with exotoxins. We have different types of bacteria that produces different types of toxin, and these toxins have different types of mechanism. So let's start. First of all, we have a type of toxin that inhibit protein synthesis. Now, different examples these includes. If we talk about Corynebacterium, diphtheria, this produce diphtheria toxin. What is the mechanism of this diphtheria toxin? That is inactivate elongation factor EF2. EF mean elongation factor through ADP ribosylation. And we have Pseudomonas aeruginosa that produce exotoxin A. So Pseudomonas aeruginosa produce exotoxin A, which is also protein synthesis inhibitor. Now Corynebacterium diphtheria produced diphtherial toxin that cause pharyngitis. Remember, and that is with pseudomembranes in the throat. If you have seen white pseudomembranes and severe lymphadenopathy, that is called bull neck and myocarditis. So these problems are caused by Corynebacterium diphtheria toxin called as diphtheria toxin. Simple name. Remember, pseudomembrane in diphtheria, white membrane in diphtheria, gray type membrane. Then we have Pseudomonas aeruginosa that produce exotoxin A and cause host cell death. ADP ribosylation, what does it mean? Add ADP ribose to a protein and the protein become dysfunctional. So this is ADP ribosylation. Then we have Shigella species. This is a bacteria that produces toxin Shigella toxin A. And what is the mechanism? The mechanism of Shigella toxin A is inactivate 60S ribosome by removing adenine from ribosomal RNA. So from ribosomal RNA, adenine is removed and this inactivates 60S ribosome by this mechanism. What disease it causes? That it is, and as well as enterococci hemorrhagic E. coli damage the GI mucosa causing dysentery. Remember dysentery. Toxin mediated injury and cytokine release causing hemolytic uremic syndrome, that is in short form called as HUS. Prophylactically in enterohemorrhagic E. coli serotype 0147 the H7, this one is specifically. Unlike Shigella, enterohemorrhagic E. coli does not invade the host cell. So just like Shigella, invade, but EC does not invade the host cell. So these are different types of toxins that inhibit protein synthesis. We have next type of toxin that increase fluid secretion. In the example, we have enterohemorrhagic E. coli. This is eat labile, that it is sensitive to eat toxin. Remember LT, labile toxin. Overactivate adenyl cyclase that increase the CAMP level and increase chloride secretion in the gut and H2O efflux is increased. That cause watery diarrhea, and labile in the here mean adenyl cyclase and stable on the ground mean guanylate cyclase. So this is a mnemonic. Then we also have eat stable toxin that are ST. The mechanism is that it overactivate guanylate cyclase and increase GMP, and this is decrease reabsorption of NaCl and H2O in the gut. So NaCl and H2O reabsorption is decreased, that also cause diarrhea. Bacteria that increase cyclic EMP include Vibrio, Bacillus anthracis and Bacillus pertussis, and E. coli, Bacterial-pertussis. So these are the eat stable. Then we have Bacillus anthracis, and that produce a toxin called as anthrax toxin. So, and this mimic adenyl cyclase increases the cyclic AMP level, and this is likely responsible for characteristic adenomatous border of black scar in the cutaneous anthrax. So this toxin is responsible for black scar in the disease that is cutaneous anthrax, and that have edematous borders. Then we have Vibrio that produce a toxin called as cholerae, which activate adenyl cyclase, increasing the level of cyclic AMP and permanently activating GS protein. This called voluminous rice watery diarrhea. So this is important for the exam purpose. So Vibrio cholerae is a bacteria that produce a toxin called as cholerae, and that cause voluminous rice water diarrhea. Then we have toxins that inhibit phagocytic ability, that include Bacterial-pertussis, producing a toxin called as a pertussis toxin. This also increase the level of cyclic AMP, inhibiting G inhibitory proteins. And the most important with respect to exam is this thing, that it causes whooping cough, and in this cough we have child cough on expiration and whoops on inspiration. and it can cause unread day cough in adults associated with pertussis emesis. So it can cause unread day cough in adults, and this is associated with pertussis emesis. So these things are also important for the exam. Then we have enzymes, toxins that inhibit, and that we have exotoxin that inhibit release of a neurotoxin that include Clostridium tetani, and this bacteria has a toxin that are called as a tetanus plasmin. Then we have Clostridium botulinum produce botulinum toxin. So both are proteases that cleave snare soluble NSF attachment protein receptor, and that is the full form. A set of proteins required for neurotransmitter release through vesicular fusion. Toxin prevent release of inhibitory and glycine. So the Clostridium tetani that prevent the GABA and glycine release, and we know that GABA is a glycine are inhibitory. So when GABA and glycine which are inhibitory are not released, then excitatory activities will occur. And you also remember that these are produced by Lenfro cell in the spinal cord. So GABA and glycine inhibition from Lenfro cell in the spinal cord is inhibited. So if we have inhibited GABA and glycine, then we have increased neuronal activities. So spastic paralysis, rhesus donicus, trismet, that is also called lagja, and epistetonus, these things will occur. So this will be the final result of Clostridium tetani. And Clostridium botulinum, this bacteria produce botulinum toxin as we discussed. In fact, cause infant botulinum. Caused by ingestion of spores from cell raw honey, toxin is produced in vivo. So in case we have used, if we give honey to a baby or he get it from the soil, so he can ingest botulinum toxin of Clostridium botulinum that can cause infant botulism. It can be foodborne botulism, and that is caused by the ingestion of a preformed toxin from a canned food. So canned food can also cause botulinum toxin ingestion problem. Now what is written over here, let me discuss. A toxin have two components, A and B. Toxin have two components, one is A and one is B. The active component is toxic. This is the active component and this is toxic, and A for active you can write, and B for binding. This is for binding to a cell. So if we have a cell, let's say this is a cell, and we have toxin, this is a toxin. It has two components, B. The B component of the toxin bind to the cell, and the A component of exotoxin then cause the problem, which is the active component. So A and B mean this. So exotoxin has two components, A and B. A is toxic and A for active. B is for binding of exotoxin to cell surface and B for binding. So you should remember this thing. Then another bacteria that and another mechanism that is lyse cell membrane that have Clostridium perfringens. In case of Clostridium perfringens, it produces alpha toxin and phospholipase, that is lysigenase, that degrade tissue and cell membrane. So we have Clostridium perfringens that is alpha toxin, and the mechanism is that it is phospholipase and degrade the tissue and cell membrane. When degradation of phospholipids occur, and that cause myonecrosis, necrosis of the muscle, and cause gas gangrene. So with respect to exam and clinical practice, again this point is very important. Myonecrosis or the gas gangrene is caused by degradation of phospholipids, and the most common cause is Clostridium perfringens, and that produces a toxin which is called as a alpha toxin. So gas gangrene also it can cause hemolysis, double zone of hemolysis on blood agar is seen. So remember the two things, hemolysis and gas gangrene, more specifically gas gangrene, which is very important, is you should remember. Then we have Streptococcus pyogen. Streptococcus pyogen produces a toxin that is streptolysin O. This protein that degrades cell membrane. So this streptolysin O also degrades the cell membrane. Red blood cell lysis contribute to beta hemolysis. Asked antibodies against toxin ASO used to diagnose rheumatic fever. Do not confuse with immune complex of post-streptococcal glomerulonephritis. So in this case we have antibodies against toxin that is produced, and for the diagnosis we detect these antibodies in the rheumatic fever. Then we have some super antigen that cause shock, Staphylococcus aureus, toxic shock syndrome, cross-linked beta region of the TCR to MACH, class 2. We have class 1 and class 2 MHCH on the antigen presenting cell outside of the antigen binding site. Manifestation of toxic shock syndrome is fever, shock, and rash, and other toxin cause scalded skin syndrome, which is exfoliative toxin, and food poisoning can also be caused by heat stable enterotoxin. So toxic shock syndrome is caused by Staphylococcus aureus. You must remember this thing. If someone asks you the toxic shock syndrome cause, then you should say Staphylococcus aureus. Then we also have Streptococcus pyogen. Streptococcal pyogen that we discussed above produce erythogenic exotoxin A also, an overwhelming release of interleukin 1 to interferon gamma and T1 effector alpha, and that result in the shock. Toxic shock syndrome, which is also again in fever, rash shock, and scarlet fever. So toxic shock syndrome and scarlet fever is caused by the Streptococcus pyogen. And the toxic shock syndrome is seen in the female that use tampons. So in those female it is seen. Then we have endotoxin. In endotoxin, as we discussed, it is a lipopolysaccharide. It is found in the outer membrane of gram-negative bacteria that we have discussed. Both cocci and rods can be, and this is composed of O antigen plus core polysaccharide plus lipid A. The toxic component of endotoxin is lipid A. So the composition is O antigen, core polysaccharide, and lipid A. And among the three, the toxic component is lipid A. If we talk about a Neisseria that have lipo-oligosaccharide, so we say that lipopolysaccharide is the composition of endotoxin, including also is a whole O antigen, core polysaccharide, and lipid A. But if we say Neisseria, then we have lipo-oligosaccharide. So released upon cell lysis or by living cell by bleb detaching from outer surface membrane. Versus exotoxins, which is actively secreted. So this thing we have discussed, that endotoxin is produced only by bacteria that is gram-negative and when it lysis occur, but exotoxin is secreted. The main effects is that it has is macrophage activation, complement activation, and tissue factor activation. So these are the three main: macrophage, complement, and tissue factor activation. These are the three effects of endotoxin. Then endotoxin have these effects: edema, nitric oxide, deacid death, outer membrane, T1 alpha, antigen, extremely this thing, interleukin 1-6, neutrophil, chemotaxis, and shock. So if we discuss this is the endotoxin that macrophage has three functions: macrophage, complement, and tissue factor activation. Tissue factor activation cause coagulation, cascade, and that causes DIC. Then we have complement activation cause C3A and C5A. C3 cause histamine release called hypotension and edema, and that cause neutrophil chemotaxis, C5A. Macrophage activation, interleukin 1 and 2, which cause 6 cause fever, interleukin 1 and 6 cause fever, T1 necrotic alpha cause fever and hypotension, and nitric oxide cause hypotension. So these were the different types of exotoxin as well as endotoxin.